Hello. Thank you to our hosts, first of all, for inviting me to speak to this very unusual, specially selected group of attendees. I look forward to meeting you all on November 12th. I thought you might like to see where I'm speaking from. The X marks Calgary, Alberta, just north of Montana. And this is what Calgary looks like on a good day, Rocky Mountains in the back. And notice the lovely river that the city sits on because I'm going to mention it later. And this is who is speaking to you in case you wanted to have a visual image. So I'd like to start out by saying a few words about why I selected this title, Mind and Metabolism. Through my own academic lifetime, we have watched the rise and fall of the chemical imbalance concept of mental disorders. It really originated in the 1960s and 70s, and you may know it as the monoamine hypothesis. And of course, there's been a lot of research on neurotransmitters, the monoamines, since then, and very growing and developing field all the time. But I think what everyone expected was that the ensuing research would support the theory of chemical imbalance as the etiology of mental disorders. And that really hasn't happened. So now we have comments such as this appearing in 2011. It's kind of interesting to think of the whole notion as an urban legend. But the point I want to make is that as people are still sweeping the bits of the chemical imbalance theory into the dustbin, we continue to ask that age-old question, what causes mental illness? And unless you believe that psychiatric symptoms are manifestations of possession by evil spirits, in which case we should all become exorcists, then I personally believe that you must attend to brain metabolism and the role of nutrients. This is the article that I think led Mitch and Eric to invite me to speak at this meeting. And we began the article by saying that we are now living in a transformational moment in history. It might sound a little grandiose, but I actually think it's true because of the re-emergence of understanding of the role of nutrition for mental function. I say re-emergence because there's a long history of this idea, and I have one 45-minute lecture on the internet all about the history, tracing it back 2,800 years, but I don't have time to go into that today. So the first part of my talk is going to focus on the four components which we covered in that article, which are inflammation, the microbiome, oxidative stress, mitochondrial function. And then the second part will show you that all of these roads lead to nutrition, and that's the point overall. And by nutrition, I want to put it right out there in front. I'm not talking about herbs or exotic things, but the basic essential nutrients that frankly my generation has been taught to take for granted ever since at least World War II. So it seemed like this was a good place to put my disclosure slide, and I want to make a couple of very important points here. In the second part of my talk, when I talk about nutrition, I will be mentioning several companies, so I'll list some of them for you. I've been studying the concept of broad-spectrum micronutrient treatment, and by the way, I use the term micronutrient in this lecture to refer to minerals and vitamins. But the fact is I can't manufacture formulas myself, so I study formulas that other people have developed. But I've never been affiliated with these companies, and in fact the people there know they are not permitted to buy me even a cup of tea, which is actual fact. And this is true of all the researchers in Canada, the US, and New Zealand who are studying the formulas developed in Alberta in particular. And yet, and this is why I'm kind of discussing this at length here, I still regularly receive post-lecture audience feedback basically saying that I'm lying about this. I used to get angry about it because nobody likes to be called a liar, but now I kind of feel sad because I'd just like to point out that the people who don't believe that this is true are people who have been so schooled in the corrupted method of having pharmaceutical companies pay for clinical trials and give honorarium and all kinds of stuff, resulting in the distortions of the scientific data in mental health, as shown in the recent disclosures like about Paxil and Celexa, that they really don't understand that there's any other way to do research, which is very sad. But frankly, it has nothing to do with me. So that's my disclosure, and there's a disclaimer on this slide because I am only talking about nutrition, but it doesn't mean that I don't think there are lots of environmental causes outside of nutrition, like early child sexual abuse, etc., that are relevant topics. It's just not what I'm covering today. So what about those four concepts? I'd like to point out that the lay public has developed the tendency to refer to inflammation as if it is a terrible thing. But the fact is that inflammation is a very good thing. It's a healthy response to infection. We understand a lot of the components very well, and we know that that's how we fight off various kinds of diseases. However, the low-grade chronic excessive inflammation, which was identified over 25 years ago, it's kind of amazing how long knowledge translation takes, has shown that it is detectable in people with a variety of psychiatric symptoms. So the scientific question for the last 25 years has been, does inflammation cause depression, or does depression cause inflammation, or is there a third factor causing both? The answer to that question is quite complex beyond the scope of this talk, but I thought I would just show you a sample kind of study. People have used a variety of biomarkers as indication of excessive inflammation: C-reactive protein, homocysteine, the cytokines, especially interleukin-6 and TNF-alpha. And one example of a study is the Cooper Center study with about 12,000 people, showing that elevated homocysteine levels were associated with a 26% increased odds of scoring in the depression range on the Center for Epidemiologic Studies depression scale, even after controlling for variables that they know were likely to influence both inflammation and depression, like age, sex, physical activity, and so on. I'd just like to point out that studies have also reported increased biomarkers of inflammation in other disorders beyond major depression, bipolar disorder, schizophrenia, and so on. So if you would like to explore the literature in this field further, I strongly recommend you look at the article published a couple of years ago by Michael Burke et al. Wonderful title. So depression is an inflammatory disease, but where does the inflammation come from? And in the article, by the way, they point out that they're not talking about only depression, they're talking about all the common mental disorders. Where does the inflammation come from? This I just wanted to summarize quickly because it ties in with this talk. So based on an extensive review of the literature, there's certainly a lot of psychosocial stressors, poor diet, you can guess why I put that in red, physical inactivity, obesity, smoking, altered gut permeability, which we'll be talking about, and so on. I'd like to point out that this is a really optimistic message because these are mostly modifiable risk factors. Now, I'm going to move on to the second factor, the microbiome, and then I'll tie it into inflammation for you. Once again, this is a very old idea. I could show you some wonderful descriptions dated to the 18th century of the relationship between gut function and mental function. In other words, if your gut isn't working well, irritability, depression, and so forth. It's amazing how this has been known for years and years. You all know, because otherwise you have to have been asleep for the last few years, that the microbiome is a hot topic for very good reason. Most of the cells in and on our body are not our own. They are in other microbes, and those microbes are mostly, though not exclusively, in our gut. And the term dysbiosis refers to an imbalance in the gut microbes, not that anyone knows what a perfect balance is for any individual person, which makes probiotic treatments very complicated indeed. But we certainly know that dysbiosis is associated with various kinds of mental dysfunction. So for purposes, again, of this talk, what are the causes of dysbiosis? Well, of course, broad-spectrum antibiotics. Once again, why would I put diet in capital red letters? As you can guess, it's very, very important. The hygiene hypothesis, psychological stress and the HPA axis, and early life stress, etc. How do these two things that I'm talking about relate to each other, inflammation and the microbiome? There have been a variety of mechanisms hypothesized, but I'd like to focus again on an old idea that has been called the leaky gut for at least 20 years. I don't know when the term was first coined, and studied by people who call themselves specialists in functional medicine and naturopathic doctors. Now it tends to be called bacterial translocation, and I'm going to show you a picture in the next slide, but I just need to explain that our gut mucosal cells form an almost impermeable tight junction barrier to keep microbes in the gut and not leaking out when they shouldn't. Keep all those little critters that are responsible for breaking down our food, absorbing nutrients, and in some cases actually synthesizing some vitamins. And these protein strands are, I don't know exactly how they're built. Maybe that's an important question to explore for someone looking for a term paper. But these protein strands that form the tight junction barrier sometimes break down, are not built properly, and these bacteria do leak out, and they cause inflammatory reactions in the immune system, which is thought to be a very important mechanism for affecting mental function. So this is a little diagram so that in case this is a new concept for some of you, it's showing it's a kind of a cartoon of the lumen of the small intestine showing the tight junction in the first few lines to the left, and then a loose junction where the protein strands are not well developed or have broken down, and there is no control of absorption of nutrients and also the location of the bacteria, and you can see some of them leaking out. I apologize for a couple of the slides that I have. It would be lovely to have a pointer, but we haven't been able to get it to work. So inflammation certainly is one causal factor in mental health problems, as are gut dysbiosis and some of the other things we've been mentioning. But another one is, surprisingly, the act of breathing. Oxidative stress is the third factor I'd like to mention, and it's another term that's thrown around as if it is all bad, but it isn't. Our normal metabolism generates pro-inflammatory molecules. If you breathe, you are generating reactive oxygen species. If you have metabolism happening, in other words, if you're alive, ROS is being generated. And by the way, the word reactive refers to the unpaired electrons. The problem occurs when oxidative stress is out of control. And then there's damage to cell structures, DNA, RNA, etc. We know that oxidative stress can contribute to apoptosis, is detectable at higher levels in neurodegenerative diseases, and also possibly in mental function in general. One of the interesting things about reading articles on inflammation and oxidative stress is that the publications always seem to end at the end of the discussion section with some speculation about medications that could be developed to treat excessive levels of inflammation. And it's true that even though inflammation is helpful, as this slide says, helps us fight infections, but too much of a good thing is not good. And so one can see why people might think about looking for medications to fight this. However, what troubles me is, why are we ignoring the normal mechanism that our bodies can use to manage excess inflammation and oxidative stress? Why aren't those discussion sections talking about the normal way we fight inflammation? A healthy body has a number of ways to do that, and in order to explain further, we have to go back way back in history, several billion years ago. Billions of years ago, early eukaryotic cells, which characterizes multicellular organisms like us, engulfed a primitive bacterium that was like our current mitochondria. The result was a complex composite cell that produces energy, which, as you know, is ATP. So we carry these primitively derived bacterial organisms in every cell in our body, and they work for us. Or, as a philosopher might ask, are we working for them? But that's another story. Think about that one. Anyway, this change apparently gave us a competitive advantage in evolutionary terms because we can produce our own energy to make all of our organs function. Let's talk a little bit more about mitochondria. You may be very familiar with the work on the mitochondrial genetics because that's such an exciting area right now, but I am not going to go into that because what's important for this talk is the production of ATP, because that relates to nutrition and mental function. ATP is produced via two chemical processes, the Krebs cycle, also known as the citric acid cycle, and the four-step electron transport chain, which is part of oxidative phosphorylation. Now I have a cartoon here that I'm going to have to verbally direct your attention to different parts of it since I don't have a pointer, but I'll try to be very simple about it so you can follow me. The main large oval green thing in the center is the mitochondrion, but it is shown on the left, upper left, in a yellow circle as being part of the cell. And you can see the nucleus there, and there are 3 mitochondria. I want you to notice the tiny little yellow circle that is feeding things into the cell, which go to the mitochondria to make it work. And it's a capital letter N. It stands for nutrients. So this is where nutrition enters into the Krebs cycle. And that's what you see with the purple K in the mitochondria. And that's showing you the Krebs cycle, which results in the generation of free electrons. What happens next? Those free electrons have to be shuttled into the electron transport chain, I keep saying change, sorry, electron transport chain, ETC. And the thing that shuttles them there because that occurs in the inner membrane of the mitochondrion, are nutrients. So nutrients carry those free electrons over to the inner mitochondrial membrane, and that's where we have the 4 steps. I haven't drawn them all here, but 4 steps that are very well worked out, which are part of the ETC. And there are nutrients involved in every one of those. And as you can see then, coming off of the ETC in the upper right, you can see the production of ATP and in red, the ROS. So our natural defense against inflammation, dysbiosis, and oxidative stress, the 3 previous concepts we were talking about, is in the mitochondrion. It's the production of ATP. And interestingly, all mitochondrial function is dependent on nutrients, possibly all nutrients. I'm not certain of that, but one of my former students, Kevin MacLeod, put together a list of nutrients for which he could find defined roles in ATP production. And I'm going to just skip through these quickly. You can go back and read them later if you want. But the first slide shows a bunch of B vitamins. They all have known roles in ATP production in the electron transport chain in particular. Most of them vitamins B, C, D, E, and lithium. On the second slide, on the third slide, we get to some of the dietary minerals and also omega-3 fatty acids. So we stop there. I mean, it just seemed like probably all nutrients are functioning to help cells do their work, mitochondria to produce ATP so that we can fight excessive inflammation and excessive oxidative stress. I'd like to comment, where is ATP required? All cells, every single cell in our body requires it in all functions. But the 2 organs which are the greediest, that need the most ATP, are the brain and the heart. And you might not know this, but even though our brain is only about 2% of the cellular mass of our body, at least 20% of the energy from mitochondria that is generated goes to the brain. So it's the biggest energy guzzler in our body, or maybe tied with the heart, and it makes huge demands on mitochondrial function. So I think that I'm done talking for now about the emerging field of nutritional mental health based on those 4 topics. I think, as you can see, all roads lead to nutrition, and that's what we're going to talk about now. So what can I say in only about 20 minutes that will convince you of the breadth and depth of this topic? I think the first thing I can tell you is about some other resources which cover it more fully if you want to read and listen to more. There's a TEDx talk by Julia Rucklidge on YouTube. I'll be talking about her work later. There are 3 CME accredited lectures at maddenamericacontinuingeducation.org. You can watch for free if you're not looking for CME credit. You can email me to get on my email update list. I send out about 3 emails a year just to update people on broad spectrum micronutrient treatment of mental disorders. And you can look into ISNPR, which is a relatively new international society devoted to nutritional psychiatry research. And more importantly, Their third meeting will be here in North America, and it's going to be practically in your backyard in Bethesda next summer. I think it's end of July and first couple days of August. You can find out about it at isnpr.org. And you can find a lot more research on nutrition and mental health at that site too. So what I'm going to do is summarize 3 kinds of research linking nutrition to mental health and then talk about policy implications. And the 3 kinds of research are correlational, prospective studies, and treatment studies. The correlational studies are pretty easy to summarize because they all find the same things. There are around 12 large nutritional epidemiologic surveys. None from North America of this magnitude. They're from Australia, Spain, the UK, and Japan. But as I say, they all find the same thing. People who eat unprocessed, who cook their food and eat basically a more Mediterranean, prudent diet, have a lower rate of mood and anxiety symptoms than people who eat a Western or processed diet, sometimes called the standard American diet, which has the unfortunate acronym of the SAD diet. They have a higher rate of mood and anxiety symptoms. Just to make sure you know what I mean, this, of course, is an unprocessed food diet. I don't have whole grains here, but this is fruits, veggies, and fish. And here's a SAD diet. The processed foods, which a recent survey in Canada showed constitute more than 25% of the caloric intake of our teenagers, which is scary. Anyway, the correlational data are interesting. They're really pretty consistent, but you should be sitting there saying this woman is crazy if she thinks that proves causality, because we all know that people who have mood and anxiety symptoms seek processed food, and that's absolutely true. And that's why we also need prospective longitudinal studies, and we have them. So there are several, not 12, but there are several, and I usually talk about the one from Spain because I think that they had the longest time frame, and I think they did the best statistical kind of analysis. What they did is they did a semi-quantitative food frequency questionnaire with people and asked how much they ate in the category of commercial baked goods, which they defined as muffins, donuts, and croissants. They also asked how much they ate in terms of fast foods, which in Spain was referred to as hamburgers, sausage, and pizza. Those categories were added together to form an index of an unhealthy processed food diet. And they divided people into thirds, those who ate the most of that sort of thing, the middle amount, and the least. And then they sat back and waited. I should have mentioned that all the participants in this study, this was not a clinical study, they were volunteers, university people, so-called normal people, not symptomatic in terms of depression and anxiety. So they tracked these people over time to see who would end up with a diagnosis of depression. And this is the result from their sample of about 9,000 people, on average 6.2 years later. The bar on the far left is showing you arbitrarily set at 1 risk of depression, the people who ate the least processed food. The second bar, those who were in the middle third, and I think it's so interesting that there's no difference between those two. But then the people who ate the most of the items on the unhealthy diet list clearly were at significantly elevated risk for depression. So am I saying that muffins cause depression? I hope you don't go away with that interpretation. Certainly eating some muffins and other kinds of processed foods, the middle third doesn't seem to increase the risk for depression. All that this study is showing, and it's such an important finding, is that with every bite we're making a choice. And if most of your bites, or a very significant number of your food ingestion, is in fast foods, you simply are not getting enough of the other nutrients. So the obvious solution is tell everyone to eat better. And yes, that's absolutely the first thing we need to do, and we need to teach people to cook. In fact, I would say that teaching people how to cook is one of the most revolutionary things a mental health clinician could do. And they didn't teach us that in grad school, did they? But it's important to teach the population to eat better, without a doubt. There is a study that hopefully will be published this year that I'm somewhat familiar with, where a group of people with major depressive disorder were taught to eat better, and about 1/3 went into remission, which is fantastic. But what about the other 2/3? And there's reason to think that just teaching cooking is not going to solve as much as also using nutrients in pill form. And so now I'm going to talk about treatment. What's the evidence for taking nutrients in pill form? Back in 2007, my colleagues and I published a review article in Psychological Bulletin called Vitamins, Minerals, and Mood. We reviewed the literature from when research of treatment in the modern scientific era, research of single nutrient treatments really began in earnest, and that was in about the 1920s. And if you read some of the studies back in the 1920s, 30s, 40s, you would think that people had it all sewed up. There were some major transformations when groups of people were given calcium, or when they were given manganese, or when they were given other nutrients, B vitamins. So it's interesting to see how much research there was. But, the results were always, in terms of group data, rather modest. The strongest evidence was for a few of the single nutrients shown as D and E. But then when you reach the year 2000 and people started to study multi-nutrient formulas and even bigger, broader spectrum formulas, then we started to get much larger effects. And I want to show you this slide because I think it might explain to you why single nutrient research has been disappointing. In fact, I've been told repeatedly that this is the most important slide that I ever show, and it's the only one that people even remember, which is humbling, but okay. I'm pleased that it's being used in a number of schools in Alberta to explain to kids why they need to really eat broadly and make wiser food choices. And keep in mind that we already know that our mitochondria need virtually every nutrient to function. What this slide is going to explain to us is how very directly nutrients can impact neurotransmitter availability and function. So here's the history to this slide. I need to spend a minute on it. About 15 years ago, when people were making me feel as if I were studying magic, I literally was told that vitamins and minerals can't affect brain function. Why was I studying this kind of thing? I searched the internet to understand more biochemistry than I had been taught in my training, and I decided to really look into the neurotransmitters and what kinds of molecules were needed to synthesize and break down neurotransmitters. I made this slide based on two criteria. First of all, I picked serotonin because even 15 years ago, lay audiences would know that word. Everybody knew what serotonin was. And secondly, it had to fit on a single PowerPoint slide. Beyond that, as I'm sure most of you will recognize, this is a very tiny, tiny portion of the complex metabolic pathways in our brains. So what can we see here? These are the steps going from tryptophan to serotonin, and then there's one breakdown step also. And everywhere along the way, enzymes make these steps work. And I have not put in the names of the enzymes that we get too crowded. What I did do is I asked the question for each arrow, what nutrients are involved in the cofactors or coenzymes that enable those enzymes to work optimally? So here we have vitamin B6. In fact, pyridoxine is in a number of steps in this little snapshot. Here we have a dietary mineral, copper, another place for copper. Here we have a place for iron, and here we find, another second place for iron, right? And here, molybdenum. The enzymatic reaction breaking down serotonin requires a certain amount of molybdenum, which is a trace mineral in a lot of our vegetables, for optimal function. So you have to ask yourself, given that so much of the SAD diet, and we shouldn't just call it the standard American diet, believe me, it's the standard Canadian diet too, it's processed food. Where are these nutrients coming from which are required for adequate pathway function in a human brain? Around the same time, I read this important article by Bruce Ames, the famous neurobiologist. He reviewed 50 known mutations that result in unusually high amounts of nutrients required as cofactors for enzymes to work optimally. And this happens because the mutation seems to affect Micheli's constant, which is the measure of binding affinity for an enzyme, or by an enzyme for a coenzyme, or also called a cofactor. This particular summary, which I recommend to you, was limited to about 50 mutations known that cause known human physical disorders. In other words, no mental disorders. And it was also limited to coenzymes involving vitamins, not minerals. So it's not even a complete story. But the message was clear. In all 50 examples, simply feeding people more of the vitamins that they needed restored adequate enzymatic function and improved those sluggish pathways. So, I wondered, could some forms of mental illness reflect sluggish metabolic reactions? In other words, could mental disorders be inborn errors of metabolism affecting Micheli's constant, resulting in enzymes requiring kind of a flooding of micronutrients so that cofactors would be in sufficient quantity to optimize those metabolic steps? And I thought this was a novel idea. Turns out that Linus Pauling, the very esteemed Nobel Prize winner, had said it all many, many years before. As this is kind of a summary of a couple of comments he made, but what he was proposing was that the genes for mental illness are likely the genes that regulate brain metabolism of essential nutrients. And I think we're eventually going to know that that is absolutely true. Not that we can prove it yet completely. Now, I have one more metabolic pathway slide I want to show you because I'm not talking about omega-3s. Very important area, very important that we all take it, that we eat fish, important for our heart and our brain. We all know that. I don't follow the literature as closely in terms of mental health, as closely as the literature on minerals and vitamins, in part because I think it tends to perpetuate the single nutrient approach, which I believe is inferior. And this kind of shows why just focusing on a single nutrient like the omega-3s in isolation doesn't make a lot of sense. On the left, you have omega-3 pathway going down to, you'll spot EPA and DHA that I'm sure you'll recognize, and that's an anti-inflammatory pathway, broadly speaking. And on the right, omega-6 going down to the metabolic steps lead to arachidonic acid, and that's a pro-inflammatory pathway, generally speaking. And in the middle, you'll see some of the enzymes listed. And then you'll see these little yellow arrows, and those I have superimposed on this figure, which is freely available on the internet. And what I've done is I've just pointed out where cofactors involving standard micronutrients have been identified. So for example, the top one requires vitamin C and some B vitamins, magnesium, zinc, and so on. So the point I want to make here is you can take all the omega-3s that you want. If you are not getting optimal levels of the micronutrients that are required as cofactors, even taking those pills and eating your fish, you might not be converting it adequately into the end products that we know are very important for brain function. Now, one more thing about single nutrients, because of some interesting work that came out this year. The use of single nutrients to boost the effect of medications is also kind of an old idea. And I'll just show you three references. Back in 1992, Iris Bell showed she could amplify the effect of tricyclic antidepressants with B complex. And then in 2008, the Harvard Mental Health Letters summarized some literature reviewing folic acid, SAMe, and omega-3s and concluded that all of them boost the response to psychiatric medication. This year, however, Jerome Saras from Australia et al. Published in the American Journal of Psychiatry a meta-analysis that showed that there was good reason to think that certain individual single nutrients boost the effect of medication. And they included, again, SAMe, methylfolate, omega-3s, and vitamin D. So this is great to know. First of all, it fits the model I'm presenting to you. And if your goal is to enhance medications, it's good to know. But didn't we just see lots of evidence that we've evolved to need a broad range of nutrients, both for mitochondrial function and for brain metabolism? Where's the molybdenum in these studies? No one's going to study that in our lifetime. Why are we focusing on just single nutrients? And by the way, do we have to focus on enhancing medication? Or could we, in fact, put some people on micronutrients so that they would never need medication? And that's the research that's been going on since the year 2000. Now, my own work has been on Alberta formulas developed in Alberta. I'm going to refer to them as the Alberta formulas. There's more than one. And there are about 30 peer-reviewed publications on them. They are broad-spectrum formulas, and we use them instead of medication most of the time. And if you would like to see the list of those publications, I'd be happy to send them to you. I'm going to present you with just a bit of the research on those formulas. There is also research on other multi-nutrient formulas that are not broad spectrum. They might be like just B complex or B complex and a bit of omega-3s or something like that. But all of them are really quite exciting and positive. I'm focusing on the Alberta formulas because it's my area. I usually review this literature in terms of levels of evidence going from case studies up to randomized controlled trials, but without sufficient time, I'm just selecting a few things here as shown in blue. So I need to tell you about the Alberta formulas. There are approximately 30 minerals and vitamins. They're above RDA levels. They start out with one company, True Hope, but they split into two around 3 years ago. It's always very hard to explain. So now there's True Hope and Hardy Nutritionals, and then True Hope partnered with a third group called Q Sciences. You can look at their websites and read about them, truehope.com and hardynutritionals.com. The point is, I'm not affiliated with them. I don't promote their products. You will see all of us researchers on their websites, all of us from the US, Canada, and New Zealand. But those are not endorsements. It's just that when any of our publications enter the public domain, any company can put them on their websites. I don't really care. So here's an example of a design which I think is a very useful one, showing on-off control of symptoms and this is with one of the Alberta formulas. This was the earliest one published. It was in 2 children who entered an open label trial, but they got better on the formula, and then something changed in their lives so that they couldn't continue in the open label trial. And I'll show you with one boy. He was taking Empower Plus, which is the name of one of the broad spectrum Alberta formulas. He was a really severely disturbed child with ADHD, Asperger's, learning disability, huge outbursts, great anxiety, requiring a full-time aide in school. He did really, really well on the formula, as I'll show you in a moment. But then he changed schools for reasons having to do with availability of an opening. And so no one could conclude whether the change in schools or the formula was responsible for the improvement. So we couldn't interfere with his schooling. We simply stopped the formula, and then we did an on-off design. And I happen to know the family socially now, so I know the boy. He's actually about 30 now. So I followed him for a long time, and he has taken over complete responsibility for buying his own broad spectrum formula, and he keeps it with him at all times. So here he is on a Conners type measure going from 0 to 3, measuring mood on the left and temper outbursts on the right. And again, I don't have a pointer, so the hatchback bars are at baseline. Then the first black bar is shows how much he improved when put on the formula, but he had changed schools, so we didn't know what was going on, so we stopped the formula. And the white bar was when he returned to those very high levels of mood and temper outbursts, so we put him back on the formula, and that's the second black bar. I'll show you a much more recent use of a within-subject crossover design. This is in a sample of 14 school-age children with combined ADHD, and it was a 6-month trial, but each phase was 4 weeks. This is from Julia Rucklidge's group at the University of Canterbury in Christchurch, New Zealand. She has the most active treatment research program right now, I think, of anyone, and using broad spectrum formulas, I mean. And they were using one of the Alberta formulas. I don't remember which one actually. Heather Gordon did this as her thesis. This was pilot data in preparation for an RC. Oh, come on. Did I forget to start it? I will show you one other within subject crossover design, and this was in 14 school-aged children with ADHD, done in Julia Rucklidge's laboratory at University of Canterbury in Christchurch, New Zealand. Julia has the most active research program in the world right now. But we need to move to RCTs, and I refuse ever to talk about RCTs. And anyway, this was part of pilot work for a standard RCT, which they're doing right now. So I have only one slide on it, but anyone interested can go read it. So Heather Gordon, her student, did this pilot trial in 14 children. They're expensive. The yellow bar shows the T-score on a concentrated parent rating score for the 14 children. It's hard to do. And then the first green bar shows their improvement when taking one of the Alberta formulas for science. And then the red bar shows very few discoveries are made with RCTs, possibly none. And the red bar they actually are the gold standard. I think we can learn a lot from this kind of experimental why they were design defined by the FDA as a requirement for marketing. And I think that we have went through a phase of impoverishing our scientific literature for a while by not just the reliance on RCTs, which remember I'm saying are an important form of data, but the over-reliance on RCTs to the point where for about a decade there, I don't think you could publish a case report with a new discovery. Now we have whole journals devoted to case reports, so the pendulum is swinging back. But I want you to keep in mind that even though it is an important form of evidence, it's not necessarily the best, it's just one. Now, this slide is reflecting not just the Alberta formulas, but the world literature on multi-nutrient formulas. There are over 25 positive RCTs to date, and the only negative trials that have been reported have been in non-clinical samples, like get a bunch of college students together and see if they have any less stress on B complex or something like that. I'm going to focus first on the topic of stress and natural disasters. You'll see why. There are about 8 RCTs that have shown that micronutrients decrease stress and anxiety. The only one that has a negative result was done in normals again. I don't know how these people get funding or ethics approval. And by the way, the media always trumpets the negative results, and the public doesn't look to see, well, did they even have adequate levels of mood or anxiety symptoms to begin with to see an effect, or is there a floor effect? It's a little bit frustrating because the media loves negative results on nutrition. Now, I want to talk about 2 earthquakes and a flood. Julia Rucklidge is as I said, is in Christchurch, New Zealand. And what you're looking at here are the roughly 8,000 earthquakes that happened in a 5-month period in 2010 and 11. The first earthquake in September 2010 happened while Julia was doing some clinical trials of one of the Alberta formulas, and what she found was that the people who were currently taking formulas did much better in coping. They were more resilient to the stress of the earthquakes than people who were not taking the formulas, and she published that. Then they had about 8,000 so-called aftershocks in this short time period, which created unbelievable stress in the population, and then another massive one in February 2011 that killed a few hundred people and destroyed most of their downtown. So at that point in time, Julia and her students set up an RCT. Now this was in the general population, but a very, very stressed general population. And you can read the details here or go read the original article, but I'll just tell you a summary of the results. 3 treatment groups. You can't use a placebo in that kind of environment, of course. 1 took B complex, and they really improved in terms of depression, anxiety, and stress. Larger effect sizes for those taking 2 different doses of the broad spectrum formula. And you can see it here. On their measure of PTSD. If you look at the 3 bars on the left, that's the micronutrient group. You can look on the right for the comparison controls. And at baseline, the group receiving micronutrients, over 2/3 of them met criteria for a cutoff score for PTSD. 4 weeks later, on micronutrients, that was down to 19%. And you can see in the controls, there was really no change over that same month. And a year later, they were even better. So we then had a flood in Calgary. Remember, I showed you what our rivers looked like normally. This is what a Calgary area river looked like on one day in June of 2013. So Julie and I decided to work together and try to replicate her earthquake studies. And this is what we showed. We used a comparator of vitamin D, which is the blue line. And again, this was just a 6-week trial. And the vitamin D group, in terms of the y-axis, is depression, anxiety, and stress score. They did get better. Remember, this is a single event in a population that everyone came out to help them, not 8,000 earthquakes. So I was kind of amazed we saw any improvement, but we did. But the two groups that took either B complex or a broad spectrum, one of the Alberta formulas, improved even more than the single nutrient. So it's a pretty strong case that the broader spectrum formulas improve resilience, which we already know from all the animal data that I'm not covering here today. Somehow we need to prove these things again in humans, though. ADHD, I'm just going to present one RCT, and that's also Julia Rucklidge's, published in British Journal of Psychiatry. They had 80 adults with ADHD. Remember, the child ADHD trial is in progress. And they had 8 weeks of one of the Alberta formulas compared to placebo, classic, blinded, RCT, 8-week open label extension, and 1-year follow-up. And this is the improvement on self-rated ADHD symptoms. Improvement means that you expect to see a larger bar, and indeed you do. The blue indicates the people who got the active formula did much better in terms of improvement on hyperactivity, impulsivity, and on inattention. And this is the 1-year follow-up, which is really interesting. If you look at the yellow line, the people who stayed better in terms of all of those symptoms 1 year later were those who stayed on their micronutrients. To this audience, I would like to say that I had a big body of data on depression, but it doesn't exist yet. So I'm going to just show you what Rucklidge et al. Published in terms of depression. 1/4 of their sample met criteria for depression at baseline. And what this shows is that those who received the active formula, which is the purple bar, improved much more on mood scales. This is the Montgomery-Asperger Depression Rating Scale from pre to post than did those who received placebo. Clearly, we need more research on depression. Now, I'm going to end with some, probably just one case study and policy implications. I'll present a second case study on November 12th if there's time. I want to tell you about Andrew, who's the middle child of 3 in an intact family. He has some learning disabilities, and he had a history of lots of stress and anxiety. But when he was about 10, he got really bad, and he became so disturbed. He became psychotic with hallucinations, paranoid ideations. He thought his food was being poisoned, etc. He was convinced that he had committed adultery and murder, and so he had to pray incessantly, characteristic of an OCD kind of problem. He was treated in Children's Hospital where I lived, though I didn't know him then, and had every investigation imaginable and a long list of medication trials. You can see some of his delusions here. After 6 months, he was unchanged. They were unable to help him at all. His Children's Global Assessment Scale score was 35, and the CGAS when he was transferred to outpatient care was still 35. And in outpatient care, Dr. Megan Rodway saw him, and his parents had heard about the Alberta formulas and asked her if she would support them if they wanted to try it on this boy that we call Andrew. Dr. Rodway apparently said, "This is snake oil, but I don't have anything better to offer." So, what happened to Andrew on the snake oil? I was called in to help with the data at this time. I wasn't familiar with the family before then, but it was quite fascinating from 2008 to 2009. This is what happened to his OCD scale. The red is a 10-year moving average, his psychosis symptom scale. And then Andrew himself became able to rate his visual and auditory hallucinations after a few months on the formula, about 2 months, I think. And so this is his own self-rating of hallucinations as they went to zero. By the spring of 2009, he was doing very, very well. And by the way, he graduated high school in 2015. I don't think many children who go through childhood psychosis can say that. And I'm still in touch with him. We're Facebook buddies, and he's doing very well. He works part-time. No adverse events, etc. And it's published here if you want to take a look at the article. But here's why it's a policy issue. A health economist pulled the data. We have a single-payer system in Canada. She pulled the data on the cost of his 6 months inpatient hospitalization, compared it to 6 months immediately following of micronutrient treatment, and take a look at this. Our healthcare dollars cannot sustain the rapid increase that's happening in the current conventional treatment of mental health disorders. We should be paying attention to micronutrients for that reason alone. But also, here's a challenge for you. What about stigma? Would any of you say that Andrew has a mental disorder? Would you say he's mentally ill, or that he was mentally ill? Or would you say he inherited an unusually high need for micronutrients? Actually, there is a lot of mental health history in his extended family, and so his family may embody the concept expressed by Linus Pauling. Where do we go from here? Well, in God we trust, but everyone else needs data. But RCT data are expensive, and NIH has repeatedly rejected every proposal with a broad spectrum formula of any type. And by phone, NIMH recently told one of my American colleagues, we simply will only fund single nutrient studies. So because of that discouraging fact, I mean, I understand it. It's based on the scientific method, and I was trained in that too, but unfortunately it doesn't reflect metabolism and how the brain works. So we need more proof. We need more young scientists studying this. We need more knowledge translation. So I've set up 2 charitable funds, not to benefit myself because I'm retired. I don't set up studies anymore. One is here in the States, and it's been doing fairly well. We've raised over $200,000, and about half of that has already gone to one of the junior colleagues to do research in this area. And then I set up one in Canada, and it also has just recently scored a very large donor. And I'm pretty sure where that money is going to go when there's an ethics approval on the proposal. Why is this important? I'm going to conclude with a quiz. What do these characteristics sound like? Depression, irritability, self-harm, etc. Don't they sound like DSM criteria? Well, they're actually from a group of normal, healthy students deprived of optimal nutrition. We have known for a long time, and as I say, I could trace it back over 2,000 years for you, that nutrition is essential, and a broad spectrum of nutrients are essential for optimal brain function. But I love this quote from Marshall McLuhan: only puny secrets need protection. Big discoveries or big knowledge are protected by public incredulity. And we seem to have to keep rediscovering this simple yet very big truth. Our brains need a constant infusion of a broad spectrum of nutrients for optimal function. Thank you for listening.